Hello, this is Mohamed Abdelnaby, and today I am presenting our poster, which is on benchmarking, principal component analysis and random projection for dimensional reduction, for single-cell RNA sequencing. So both PCA and random projection are used to project high-dimensional data into lower-dimensional data. And in the case of single cell RNA, we're dealing with a really high dimensional data that we need to use dimensionality reduction techniques like PCA, Disney, UMAP, and also random projection. And in this research project, we used two datasets for our experiments. We used a targeted PBMC sourced from a healthy donor, and we also used a sorted PBMC sourced from a healthy donor. And for the dimensional introduction techniques that we used, so we're benchmarking PCA and random projection, and we did use two PCA versions, the full PCA and the randomized SVD solver PCA. In the case of random projections, we're using sparse random projections and Gaussian random projection. And we are adjusting the number of components. So the number of components are the number of projected dimensions. So let's say you originally have 2,000 dimensions or 2,000 features in your dataset, and you want to project them into 50 dimensions. So 50 is going to be the number of components. So we're varying the number of components from 50 all the way to 1,000 in steps of 50. So 50, 100, 150, all the way to 1,000. And we are also comparing and benchmarking PCA and random projection towards the downstream analysis, specifically clustering. So in this research project, we are essentially comparing PCA and random projection in terms of time complexity and time efficiency, and also in the clustering analysis. So we did use two clustering algorithms, hierarchical clustering and spherical k-means. For the assessment method, we're using the Dunn index to assess the quality of the clustering. And in terms of our observation in this research project, so first we have to talk about locality preservation and actually see if both PCA and random projection preserve locality. So as you can see here in figure 1, A and B. So figure 1A is for PCA, B for Gaussian random projection. And you get this plot if you project the dimensions to 2 dimensions and then plot the clustering. So it's a 2D plot showing component A and component B, or component 1 and component 2. So pretty much the 2 dimensions that you projected into. So you see, like, all the data points are smooshed together. You really cannot distinguish which data point belongs to which cluster. So effectively, what we would do is that we would project these data. So we would still use random projection and PCA to predict the data. And then we would use UMAP to further reduce the dimensionality and project it from a certain number up to 2 dimensions, just to visualize this nice plot right here. So here we did use PCA with 500 components. Also Gaussian random projection was 500 components, and then we further projected these 500 components to 2 components or 2 dimensions using UMAP, and we got this nice plot here, which you can distinguish the clustering So that's in terms of locality preservation. In terms of the time complexity, so figure 3, which showing all these plots right here. So each of these plots are actually coming from the same dataset. The only difference is that we did vary the number of data points in this dataset. So we did create a sub dataset or a subset from this larger dataset. Uh, so figure 3A is going to be 1,000 samples, B 2,000, C 3,000, all the way to figure 3J, which is 10,000 samples. And these are randomly sampled from this 10,000 dataset. And as we can see here, the orange line is the randomized version of PCA, the blue line is PCA4, the green one and the red one are for Gaussian, for Gaussian random projection and sparse random projection. So we can see that random projection is performing much better than PCA in terms of the time complexity. So it's taking less time for the dimensionality reduction process to complete and to get the components. And we're varying them from 50 to 1,000 in steps of 50. So effectively, we have 200 data points here in this plot. And an interesting observation that we noticed is that a randomized version of PCA is performing better up to a certain point, and then it starts performing worse. So you can see here sparse random projection, the randomized version of PCA is much effective, much faster than PCA. And then around a number of components of around 300, 400, it's starting to perform worse. And here in this plot 4, we are showing the sorted PBMC dataset in terms of the execution time. And again, same exact observation. The only difference here is that we didn't quite see that the randomized version of PCA is going to exceed the full PCA. However, if we increase the number of components further, we believe that it will exceed it. But here's the result, and again, random projection is performing much better. And for this one, we're showing the execution time versus a varying dataset size. So on the x-axis, we have the dataset size ranging from 1,000 all the way to 10,000. And we have the execution time in seconds. And we can see here that the randomized version of PCA tends to do much worse as you increase both the dataset size and also the number of components. And that's not a surprise in terms of time complexity. We know that the time complexity for both sparse random projection and Gaussian random projection is big O n by d by k. N is a number of data points, d is a number of original dimensions, and k is a number of projected dimensions or targeted dimensions. So let's say you have a data set of 1,000 features or 1,000 dimensions, and you want to project it into 50 dimensions. So the d is going to be 1,000, the k is going to be 50, which you're targeting to lower the number of dimensions into this dimension. So the time complexity for PCA is much worse than random projection. And here we're showing the Dunn index for the spherical keys clustering. So surprisingly, we did see here that the random projection, both versions, the sparse and Gaussian, are performing better in terms of the Dunn index and clustering quality than both versions of PCA. So as you can see here, we're saying that random projection is better. And also when we show it in terms of the other data set, sorted PBMC, we also seeing the same trend. However, we have to know that up to a certain point, around here, um, actually PSA starts to be better than random projection. However, it's not that big of an improvement. It's slightly better, um, but generally, even on lower dimensions, from 50 all the way up to around 600 maybe, random projection is performing better. We're also showing the same trend as you're varying the dataset size. Random projection is performing better generally, but again, as you increase the number of components and you also increase the number of dataset sizes, random projection starts to get a little bit lower than PCA in terms of the done index. And the done index, uh, the higher the better. So a higher number means better clustering. And then we're also showing the results for hierarchical clustering, and it's pretty much the same exact observation. And we can see that random projection is performing better than PCA, and also the same observation of as you increase the number of components, random projection is gonna get worse than PCA. So pretty much in this project, what we're trying or what we're aiming is that we're showing that random projection is outperforming PCA in terms of computational efficiency. We evaluated the two versions of random projection and also PCA, and we saw that across these two data sets, random projection has a much advantage in terms of speed over PCA, and also figure 3 actually showed that the randomized version of PCA is performing better in terms of speed up to a certain point and then starting to perform worse as you increase the number of components. So it's performing worse than the full PCA, which is widely used for PCA. And we also showed that random projection has the ability to preserve locality when we're projecting these dimensions into 2 dimensions using UMAP. So we're able to get this nice visualization of the cluster separated, and we're able to identify the clusters. So yeah, that's pretty much what we did in this project, and the goal was to try to benchmark uh, both algorithms, PCA and random projection, specifically in terms of time efficiency and also in clustering, which is used in the downstream analysis of single cell RNA. And we showed that PCA has a better time efficiency and surprisingly also performing well in terms of the clustering quality uh, by the done index. So that's pretty much it for our project, and thank you so much.